Assalamualaikum, I am Farno Shahira and our group will present about diagnostic immunology. As for the introduction, what is diagnostic immunology? Diagnostic immunology is a test that is used to determine the presence of antibodies as well as antigens in a patient. The term diagnostic immunology is representing the variety of diagnostic techniques which relies on the specificity and sensitivity of the bond between antibodies and the antigens. The principle of diagnostic immunology, there are a total of four primary immunological assays. The first one is the lateral flow immunochromatographic assay. It is a handheld assay device. It is simple to use and it is a diagnostic device used to confirm the presence or absence of a target analyte, such as pathogens or biomarkers in humans or animals. The second one is the enzyme-linked immunosorbent assay. It is a plate-based assay technique designed for detecting and quantifying soluble substances such as peptides, proteins, antibodies, and hormones. ELISA works on the principle that specific antibodies bind the target antigen and detect the presence and quantity of antigens binding. The third one is the electrochemiluminescence immunoassay. It is a quantitative method for measurement of antigen or antibody based on the change in electrochemiluminescence ECL signal before and after immunoreaction. It can be used to detect and quantify the presence and toxicity of specific neurotoxin based on an ACL signal. Lastly, the time-resolved fluorescence. The time-resolved fluorescence immunoassay method uses the lanthanide elements with unique fluorescence characteristics and the corresponding chelates as markers to label the target antibody or antigen. That's all from me.
Hi, I am Amin. So today I'm going to talk about the basis of complement fixation test. So this test is to determine either specific antigen or antibody is present in patient serum. So the test does not produce any visible agglutination or precipitation that can be observed with naked eyes. The indicator system is used in this test. So also the test has a low sensitivity consume more time when performing a procedure compared to the other procedures such as ELISA, IFA. It is more preferable to be used in the laboratory because it gives more advantage than this test. Next, these are the components used in the CFT or complement fixation test. The first one is the patient serum. The patient serum is burned under 56 degrees in 30 minutes. Next is the complement that usually take from the guinea pig, also non-antigen or antibody, the anti-sheep red blood cell and antibody, and the sheep red blood cell. So this is the complement fixation test. The complement in the patient serum is first destroyed or degraded in the actual test by heating the serum. After mixing with the appropriate viral antigen and also exogenous complement commonly from fresh guinea pig serum is added after the antigen antibody complex have formed. This complement attached to the complex and once fixed, it can no longer trigger lysis of the additional indicator red cell. So the anti-sheep red cell antibodies coated the sheep red blood cells are used as an indicator in this experiment. In this test, the complement is attached to an antigen-antibody complex in a positive or reactive test, and it is not free to interact with target RBC. So the RBC do not become lyse and sink to the bottom. But in the positive result of the test, in the negative result of the test, the complement is free to interact with blood cells and causing them to lyse.
Hello, I am Naziha. Now I will presenting about the ELISA, which is the acronym for enzyme linked immunosorbent assay. So here, this is the method for the ELISA test, whereas in this report, we have direct ELISA and also the indirect ELISA. So the direct ELISA is specifically for primary antibody conjugate, and the indirect ELISA is we have the primary antibody conjugate And the second one is secondary antibody conjugate, meaning that there are two antibody used. So this is the general method for the ELISA test. We have coating, blocking, detection, and the last one is read the result. Now let's move on to the direct ELISA. So this direct ELISA is an antigen is immobilized directly on the plate in a direct ELISA method, and a conjugated detection antibody binds to the target protein. The substrate will then be added, resulting in a significant signal that is proportional to the quantity of the analyte in the sample. Finally, after washing, the activity of the microplate well-bound enzyme needed to be measured. Since only one antibody is used, this technique is less specific than other ELISA techniques. This method will only be utilized to determine antibody affinity and specificity, as well as to investigate into blocking or inhibitory interaction. Now we proceed about the indirect ELISA. So just now we see that the indirect ELISA use two antibody. So an antigen, an indirect ELISA use an antigen that is immobilized on a plate and include an additional amplification detection step. First, a primary detection antibody that is unconjugated is added and bind to the particular antigen. The primary antibody is then followed by a conjugated secondary antibody directed towards the primary host species. Substrate will then be added, resulting in a signal proportional to the amount of antigen bound in the well. Finally, following washing, the activity of the microplate well-bound enzyme had to be measured. This method, this indirect ELISA method, is usually effective to measuring endogenous antibodies. That's all from me. Thank you.
Hi, my name is Dian Risalina Azira, and now I will continue about how Western blotting works. So, Western blotting is really important as it can detect a specific protein in a blood or tissue sample. The method used in this Western blotting is gel electrophoresis, which is to separate the sample's protein. A mixture of proteins is separated based on molecular weight and the type. The separated protein is then transferred onto the membrane, which is a polyvinylidene difluoride (PVDF) or nitrocellulose, where they bind to the antibodies specific to the target proteins. So due to the general high resolution capability of the gel electrophoresis and the immunoassay's sensitivity and specificity, Western blotting can identify the target proteins at the concentrations as low as 1 nanogram. Prior to immunoprobing to distinguish target proteins from off-target signals, the proteins in cell lysates were sorted by molecular mass by electrophoresis and identified using a fluorophore labeled antibody. The technique uses three elements to accomplish this task, which are separation by size, transfer to a solid support, and marking target protein using a proper primary and secondary antibody to visualize. Next, I will show this is the procedure of the Western blotting, which is the first is sample preparation, and then gel electrophoresis, then a membrane transfer, and the last one is immunodetection. Next, I will explain about the importance of monoclonal antibodies. So these monoclonal antibodies is very important, which it can reduce non-hospitalized individuals with severe disease, which have risk factors, death, and hospitalization. Then it can test for pregnancy by detecting HCG hormones in the urine. And it also can be testing for diseases such as herpes and chlamydia and HIV, which can lead to the development of AIDS. These monoclonal antibodies also can treat conditions like cancer by carrying drugs directly to the tumor cells, and it can help the immune system attack them. And the last one is it can be produced quickly despite the fact that it can be time consuming when they are made for the first time. So that's all from me. Thank you.